At first glance, the bay appeared lifeless. A vast stretch of gray-green water lay still beneath a hazy sky, its surface broken only by occasional ripples of wind. Beneath it, however, something far more troubling had taken hold. The oxygen levels had plummeted. Native species had vanished. The once thriving ecosystem had collapsed into what scientists refer to as a dead zone. And yet, into this fragile and deteriorating environment, an unprecedented decision was made. 50 million oysters were deliberately dropped into the water. To many, the move seemed almost absurd. How could shellfish, small, immobile creatures, possibly reverse decades of ecological decline? Could an organism so simple truly restore balance to an ecosystem so deeply damaged? Or would this ambitious effort become just another failed experiment in environmental restoration? What followed would challenge assumptions, reshape scientific understanding, and reveal the extraordinary power of nature's smallest engineers. But to understand how such a transformation could even be possible, one must first understand what had gone wrong. Decades earlier, this bay had been a thriving ecological hub. Its waters teemed with life, schools of fish, submerged grasses, crustaceans, and countless microorganisms forming a complex and interconnected web of biodiversity. At the center of this system was a delicate balance between nutrients, oxygen, and living organisms. However, over time, human activity began to disrupt that balance. Industrial runoff, agricultural fertilizers, and untreated wastewater flowed steadily into the bay. These pollutants carried excessive amounts of nitrogen and phosphorus, nutrients that, in small quantities, are essential for life, but in excess, can become destructive. The result was a phenomenon known as eutrophication. As nutrient levels surged, algae populations exploded. Vast blooms spread across the surface, blocking sunlight from reaching underwater vegetation. Seagrasses, deprived of light, began to die off. And as the algae eventually decayed, they consumed enormous amounts of oxygen from the water. This triggered a cascading effect. Fish and marine organisms, unable to survive in oxygen-depleted conditions, either fled or perished. The biodiversity that once defined the bay began to vanish. What remained was a fragile, imbalanced system, one that struggled to sustain life. By the time scientists began conducting large-scale surveys, the numbers were alarming. Oxygen levels in certain regions had dropped below 2 mg per liter, far below the threshold required for most aquatic species. Dead zones expanded year after year. Water clarity diminished. The ecological resilience of the bay had been pushed to its limits. And then, researchers turned their attention to a species that had once been abundant in these waters. Oysters. Historically, oyster reefs had covered vast areas of the bay. These natural structures served not only as habitats but also as biological filtration systems. A single oyster, under optimal conditions, can filter up to 50 gallons of water per day, removing algae, suspended particles, and even certain pollutants. Collectively, billions of oysters once filtered the bay's entire volume within days. But overharvesting, habitat destruction, and disease had decimated their populations. By some estimates, oyster numbers had declined by over 99% compared to historical levels. Without them, the bay had lost one of its most powerful natural defenses. This realization sparked a bold idea. What if oysters could be reintroduced, not in small numbers, but at a scale large enough to influence the entire ecosystem? Thus began one of the most ambitious environmental restoration projects ever attempted. Scientists, marine biologists, and conservation organizations collaborated to design a large-scale oyster reintroduction effort. The goal was not merely to replenish a species, but to restore an ecological function. 50 million oysters were cultivated in hatcheries, carefully monitored to ensure their health and resilience. These juvenile oysters, often referred to as spat, were then transported to designated restoration sites across the bay. The locations were selected with precision, areas where water flow, salinity, and substrate conditions would maximize the oyster's chances of survival. Then came the deployment. Layer by layer, millions of oysters were placed onto constructed reef bases, structures made from recycled shells and other materials designed to mimic natural habitats. Over time, these reefs would grow, forming complex underwater landscapes. To the untrained eye, the process appeared uneventful. There were no dramatic visuals. No immediate changes to the surface of the water. No sudden return of marine life. And this led to skepticism. Critics questioned whether the effort was worth the cost and resources. Some argued that the scale, while large, was still insufficient to counteract decades of environmental damage. Others pointed to past restoration attempts that had yielded minimal results. After all, ecosystems are not easily repaired. They are shaped by countless variables, chemical, biological, and physical, each interacting in unpredictable ways. Introducing oysters alone, some believed, would not address the root causes of pollution or nutrient overload. For months, the bay remained largely unchanged. Surface water still appeared murky. Algal blooms persisted. Oxygen levels showed little improvement. But beneath the surface, something subtle was beginning to unfold. Each oyster, anchored to its reef, was performing a simple yet powerful function, filtering water. As they fed, oysters removed microscopic algae and suspended particles, gradually improving water clarity. This process, though slow, began to reduce the density of algal blooms in localized areas. At first, the changes were almost imperceptible. Water samples collected near the reefs showed slight decreases in turbidity. Oxygen levels, while still low, exhibited minor fluctuations. Researchers noted these findings, but remained cautious. Ecosystem recovery, they understood, does not occur overnight. Yet, as weeks turned into months, patterns began to emerge. In certain regions, light penetration increased just enough to reach the seafloor. And with it came the first signs of returning vegetation. Tiny shoots of seagrass began to reappear. These plants, long absent from the bay, play a critical role in maintaining ecological balance. They stabilize sediments, provide habitat for marine organisms, and produce oxygen through photosynthesis. Their return marked a significant milestone. It suggested that the conditions within the bay were beginning to shift. Still, the transformation remained fragile. The oysters continued their silent work, filtering thousands of gallons of water each day. The reefs, once sparse, began to grow as new generations of oysters settled and attached themselves. Gradually, these structures evolved into living ecosystems. Small fish and invertebrates were among the first to take notice. Drawn by the shelter and resources provided by the reefs, they began to repopulate the area. It was a quiet return, subtle, almost unremarkable to casual observation. But to scientists monitoring the bay, it was a sign of something far more significant. The system was responding. Not dramatically, not rapidly, but undeniably. And yet, the most profound changes were still to come. At first, the change was almost invisible. The surface of the bay still carried the same muted tones, the same slow rhythm of wind and tide. To an untrained observer, little had changed. But beneath the waterline, an intricate ecological chain reaction had already begun. The oysters continued their relentless filtration. Individually, their impact was small. But collectively, 50 million organisms were processing billions of liters of water each day. As suspended particles and microscopic algae were removed, water clarity improved further. Sunlight, once blocked by dense algal blooms, began to penetrate deeper into the bay. And with that light came life. Seagrass meadows, once reduced to scattered remnants, began to expand. Their roots stabilized the sediment, preventing the resuspension of fine particles that had long contributed to the bay's murky conditions. Their leaves provided shelter for juvenile fish, crabs, and a wide range of invertebrates. This marked the beginning of a feedback loop. Clearer water allowed more seagrass to grow. More seagrass stabilized the ecosystem and improved oxygen production. Increased oxygen levels created conditions favorable for a broader range of marine species. Within a year, scientists conducting underwater surveys began to record measurable changes. Dissolved oxygen levels in previously hypoxic zones rose by as much as 15 to 25% in localized areas surrounding the oyster reefs. Water turbidity decreased significantly, with some regions experiencing up to a 30% improvement in clarity. But perhaps most remarkable was the return of biodiversity. Species that had long abandoned the bay began to reappear. Small forage fish were among the first. Their populations increased steadily, drawn by the shelter and feeding opportunities created by the reefs and seagrass beds. In turn, these fish attracted larger predators, re-establishing trophic interactions that had been absent for years. Crustaceans, particularly crabs and shrimp, also showed notable recovery. These organisms play a critical role in nutrient cycling, breaking down organic matter and contributing to the overall health of the ecosystem. Even bird populations began to respond. Wading birds and migratory species returned to the shoreline, feeding on the growing abundance of marine life. Their presence added another layer to the recovering ecosystem, linking terrestrial and aquatic environments in a dynamic exchange of energy and nutrients. Researchers were witnessing something extraordinary. What began as a targeted intervention, reintroducing a single species, had triggered a cascade of ecological processes. The oysters had not simply survived, they had reshaped the conditions of the entire environment. Yet the transformation did not occur uniformly. Some areas of the bay responded more quickly than others. Factors such as water flow, depth, and nutrient concentration influenced the rate of recovery. In regions where pollution inputs remained high, progress was slower and more variable. This highlighted an important reality. While oysters could significantly improve water quality, they were not a complete solution. The long-term success of the restoration depended on addressing the underlying causes of degradation, reducing nutrient runoff, improving wastewater treatment, and implementing sustainable land use practices. Still, the results were undeniable. Within 3 years, monitoring data revealed that certain sections of the bay had transitioned from seasonal dead zones to functioning ecosystems capable of supporting diverse marine life. Oxygen levels stabilized, algal blooms diminished in intensity, and water clarity reached levels not seen in decades. The oyster reefs themselves had grown beyond initial expectations. New generations of oysters settled onto existing structures, increasing both the density and filtration capacity of the reefs. In some areas, reef height expanded by several centimeters per year, creating complex habitats that supported hundreds of species. These reefs also provided an unexpected benefit. They acted as natural barriers, reducing wave energy and protecting shorelines from erosion. This function, often overlooked, added a layer of resilience to coastal communities facing the impacts of rising sea levels and increased storm activity. The economic implications were equally significant. Fisheries, once in decline, began to recover as fish populations increased. Local communities, many of which had depended on the bay for generations, experienced a gradual revival of livelihoods tied to marine resources. Tourism, too, saw a resurgence. Clearer waters, thriving wildlife, and restored habitats attracted visitors, generating new opportunities for sustainable economic growth. What had once been a symbol of environmental collapse was slowly transforming into a model of ecological restoration. Scientists, however, remained cautious. They understood that ecosystems are dynamic and inherently complex. While the progress was encouraging, maintaining this recovery would require ongoing effort and monitoring. Long-term studies were initiated to track changes in nutrient cycling, species diversity, and overall ecosystem stability. Early findings suggested that the bay had reached a tipping point, shifting from a degraded state toward a self-sustaining system. This concept, often referred to as ecological resilience, is critical in understanding large-scale environmental recovery. Once certain thresholds are crossed, ecosystems can begin to regulate themselves. Positive feedback mechanisms reinforce stability, making the system more resistant to future disturbances. In this case, oysters had played a pivotal role in pushing the bay past that threshold. Their ability to filter water, create habitat, and influence nutrient dynamics had set the stage for broader ecological processes to take over. The implications extended far beyond a single bay. Around the world, coastal ecosystems face similar challenges: pollution, habitat loss, and declining biodiversity. Traditional approaches to restoration often focus on reducing damage, but this project demonstrated the potential of actively rebuilding ecological functions. It raised an important question within the scientific community. Could similar large-scale interventions be applied elsewhere? Could restoring key species, often referred to as ecosystem engineers, offer a viable pathway to reversing environmental decline on a global scale? Research suggests that the answer may be yes. From coral reefs to mangrove forests, many ecosystems rely on foundational species that shape their environment. Protecting and restoring these species could unlock powerful mechanisms for recovery.